All right, sorry guys, I meant to hit the pause button and hit the stop button. So sorry about that. So let me finish the video. So we went over euchromatin and heterochromatin. So euchromatin has the acetyl groups on it. Heterochromatin has the methyl groups on it. Now, here is the difference. So on the left side here, meaning they are not acetylated, or I guess we could say methyl groups might be added to it, that's what the DNA is going to look like, all right? It's highly, highly, highly condensed. On the right side, this is the acetylated histones. The DNA is going to be a little bit looser. So this is our euchromatin. This is where our DNA will probably get transcribed so that we can get those genes activated, so that we can get those genes made into proteins, where the left, probably not. So when we go through methylation, we are adding the methyl groups to the DNA. Usually it's the cytosine that gets them, and don't forget guys, I mean, they're going to be everywhere. So if we do not have to methylate every single nucleotide in order for it to get highly condensed, when we have these areas and they're methylated, what this tells us is that that area of DNA, that gene, whatever that gene is probably not going to get expressed in that specific cell. Now, if this is like, let's say, a lung cell where that gene is not expressed, maybe in a liver cell it is, maybe in a brain cell it might be, or maybe in a heart cell it might be expressed. But in that particular cell, it doesn't really need that particular gene for that DNA based on its function. When that area becomes methylated, meaning it's heterochromatin, meaning it's not going to get transcribed and translated into proteins, this area of methylation is going to keep going through cell divisions so that the next cells also have that area methylated, and the cells after that. Reason being is we want a heart cell to have the same genes expressed as another heart cell. So if we are doing heart cell division, we want those new cells to have the same genes expressed as the previous ones. Okay, so epigenetic inheritance. And there's a video on this, guys. I will post that video for you. So the inheritance of traits transmitted by mechanisms not directly involved in nucleotide sequence is called epigenetic inheritance. Epi means above, so this is above genetics. Examples of this is like genomic imprinting, and that's a reversible form of gene inactivation that's like it's not really a mutation. DNA methylation, this is another example of epigenetic inheritance. So just what we went over. And acetylation is also epigenetic. All right, so it's above regular genetics. All right, and like I said, I will post a video for you on epigenetics, but it can be reversed, which is nice. So we can, if we see like a cell, hey, okay, maybe a heart cell now, all of a sudden, hey, I need this gene, okay? What'll happen is we can reverse that process where a mutation, yeah, it's done. That mutation is going to carry on for cell divisions to cell divisions to cell divisions. It's going to be in every single one of our cells. But the changes in chromatin, we can pass that on, and we can also change it from cell to cell, which is kind of nice. All right, so there's the video on it. I told you guys I'll post for you on Canvas. All right, we're good here. We talked about that one. Okay, so we're also going to have control elements in our operon. So we're going to kind of go back to the operon a little bit, and we're going to talk about transcription factors, and this is going to help us to regulate our process of transcription. So let's talk about last chapter. We talked about our introns and our exons. All right, and our goal is to keep the exons in and cut the introns out. All right, you would think it would be the opposite. The introns want to stay in, but no, exons stay in, introns out. All right, so we are going to have control elements that are going to be upstream from our promoter. All right, so our goal is to get the introns out. And if you remember that from last chapter, we want to get these introns out. We have our 5' end and our 3' end. All right, you remember our poly A tail there. But what we're going to be doing, like I said, we're going to cut all of our introns out. There. So when we are going to start transcription, we're going to get our RNA polymerase. That's going to go along our new strand of mRNA to make it, while looking at the DNA to make sure, okay, is A going to U? Is T going to A? You know, that format there. In order for this to happen, we got to have transcription factors to initiate and assist our RNA polymerase. Sorry, guys. Okay, so in our transcription factors, we proximal, there's two things, proximal and distal. When you talk about anatomy, proximal is like closer to the point of attachment. So like if we're talking about your arm, we would say like your bicep, tricep area is proximal in comparison to your forearm, or we could say your forearm is distal to your bicep, tricep area. So it's like, you know, proximal, and then down here is distal. But anyway, so there's different transcription factors. Some of them are going to be very close to the promoter. They're probably going to be right before. We call these proximal control factors. We are also going to have distal ones. They are going to be far away, and they're going to be a little bit different with how they're going to be used compared to the proximal ones. And as it says there, they could even be in the intron. So that's pretty far away from the promoter, because we're going to have to go through some structural genes first. So what we're going to have here is we're going to have an activator and an enhancer. And they are going to need to bind together in order for transcription to occur. So we already talked about the ocbron and regulating that. Now we're going to talk about, you know, regulating transcription a little bit further with this activator and this enhancer. All right. Skip that one. And I have a picture of DNA bending here that we'll go over in a minute. So we talked about, guys, we have to have, I'll go back here, we have to have our activator binding to our enhancer for transcription to occur. If the activator does not bind to the enhancer, transcription will not occur. So with DNA bending, and I'm going to go back to a previous picture here in a minute, but what happens if we cannot get our activator binding to our enhancer? What if we can't get it to that promoter area? So let me go back to that previous picture here. So we had our control elements really nice and close to our promoter. They were the proximal ones. They're nice and close. All right. What happens if they're really far away, either one way or the other way? We will pretend that they are way, way, way far upstream. So what's going to happen here is we are going to, and in this picture you could see it, we are going to have to do DNA bending. So we want these enhancers, these activators, to get to the promoter. So the activators, the enhancers, they're bonded together, but we need to get them over there to start transcription. So what we're going to do is we're going to pretty much make a link between our activators and our promoters. So the DNA is going to bend. Now, we can't bend it all the way, so it's like, boom, smacks up, the activators smack up against the promoter there to start transcription, but we can get it pretty close. And what we'll do is we will put this in the middle, this group of mediator proteins, to act as a link between our activators and our promoter. So the activators need to get to our promoter in order to start transcription. Over here on the left side, we're going to have a DNA bending protein. That's just going to help our DNA bend a little bit, so that way we're not tearing our DNA. Don't forget, our DNA is in a helix form, so that whole helix is bending. And then once we get the activators through the mediator proteins going to the promoter, we can then start our transcription process. Oops. Okay. So I think that's enough for today, guys. Sorry I had to make it into 2 videos today, but I think we got enough today with our transcription process and then our chromatin from the last video. So I'll see you guys next time. Have a good rest of your day.